shift slide to slide, please. I will stop my video. I will stop my video. Stop my video. Stop my video. Video off. Okay. Okay, can you see the slides? Everybody, can you see the slides?
Yes.
Okay, good. Okay. So this topic today is omic as a prognostic solution for Dr. Benny previously mentioned on the cancer. So I will focus on infectious diseases. Okay. So I hope Dr. Venu and Dr. Fama already mentioned already described you what is prognostic. So there will be a bit contradict between prognostic between cancer or non-communicable disease and also infectious diseases, because most of the time prognostic for cancer or non-communicable disease is related to the patient treatment, post-treatment, because we want to look at the compliance of the treatment and also what the effects of the treatment towards the living expenses or living expectancy of the patient. That one is for non-communicable disease like cancer, like heart attacks or myocardial infarctions or diabetes or the thingy. So other diseases, non-communicable diseases. But for infectious diseases, there will be a quite difference, because most of the time for when you are infected with microorganisms, bacteria, parasite or virus, when you are giving a drugs, most of the time the effect will be directly kill the etiology, the agents, okay, the bacteria itself, unless there will be some problem with the drug resistance parasite, drug resistant bacteria, which you need to do some modification on the treatment itself. Okay. Other than that, the prognostic for infectious disease is also related to the host itself. Okay, because the comorbid of the secondary diseases like diabetic, heart, like high blood pressures, okay, diabetic, high blood pressures, and also HIV or immunocompromised diseases also play a role to look at the prognosis, the prognostic effects of towards the infectious diseases. Okay, so before we go further, so perhaps I would like you to scan this QR code for your attendance for today's. Okay, so I give you around 2 or 3 minutes to go for this. Once you're done, just let me know. Okay. Someone from your from you, of you, if you finish, you just shout.
Done, doctor.
Done. Okay. So can I shift? Anybody still not cannot scan the QR code? I assume everybody done, eh? Okay. So if you cannot scan the QR code, you can go direct to your eLearn go to chapter 2 on TFM2 529. Then you can see the icon attendance there. Then you just click there. Then after that, you need to click on yourself. Just be aware that if you are not doing this now, so the QR code or the link for the attendance will be off at the end of this lecture, which is at 1 o'clock. Okay, so hopefully you know the time frame of your attendance, because if you are not take the attendance, there will be deduction, or there will be a marks deduction for your cost, I think. Okay. Okay, so I hope that you already knows about this kind of thingy. What is the difference between diagnosis, diagnostic, and also prognostic? Okay, so I just want to highlights again. Perhaps Dr. Venu and Dr. Fatma already mentioned this many times. Okay, but I just want to highlight again. Okay, so what is the difference between diagnostics and also prognosis? Okay. Diagnosis is mainly the process to identify the illness through the symptoms. Okay, so identify the illness through the symptoms. It means that you do the diagnostic. You want to, if you have a fever, then we just do a diagnostic to look at, is it a bacterial fever, or if this is a viral fever or bacterial fever. Okay, so when we know that this is bacterial, or viral, or parasite, so this can goes to the clinician decision of the clinician to give you a treatment. Like bacteria, definitely they will go give you a antibiotics. Okay, if the parasite, perhaps they can give you azole, okay, protozoal, albendazole, everything. Okay, so if the virus, so it will be antiviral. Okay, so when you are given these things, okay, definitely within 2 days or 3 days you will be cured. If there's no cure, then we need to do another experiment, or we need to do another checkup for actually what kind of the bacteria that you have, or is that the bacteria is not tolerant to the antibiotic, the first antibiotic, then we need to shift to another secondary antibiotics. Okay, so diagnostic will be the first process that usually we'll do for the process of the clinical processes. Okay, in order to identify what is cause of the symptoms. Okay, so prognosis usually come after diagnostic. Okay, come after diagnostic. Okay, so like the definition of prognostic is the process of predicting how soon the illness will progress. Okay, how this is a kind of prediction on how soon the illness will progress. So on prognostic, definitely we will need to know what is the species of the etiology agents. Okay, if the bacteria, is it gram negative, is it gram positive, is it Salmonella typhi, is it Klebsiella, is it Staphylococcus aureus? Okay, if it Staphylococcus aureus, is there any possibility there is a variance, or there is a virulent factors that they can produce in order to harm the host? So if we have this information, definitely we can predict that, this is a MRSA Staphylococcus, methicillin resistant Staphylococcus aureus. There will be no antibiotic can replace the beta-lactam to kill the resistance methicillin for Staphylococcus aureus. Definitely, we can predict the expectancy life of the patient. We can predict that there will be a dysfunction, the organ dysfunction after this respiratory problem for this patient, definitely, because Staphylococcus is a respiratory kind of disease. Okay, and also lung dysfunction Okay, and definitely when it becomes systemic sepsis, then it will directly go to another organ. So when there is a failure of multiple organ failure, so definitely it will go for the patient will death. Okay, so we will check we will do the predictions on these kinds of process, the kinds of the expectancy of the patient based on the agent itself. Other than agent, prognosis also can relate to the host itself, either in terms of the patient has a comorbidity in terms of the diabetic, heart pressure, heart failures, blood pressure, high blood pressure. So, and also does it has a immunocompromised diseases like infected with the HIV, infected with the TB. So, and also does the patient has a cancer? Because all these kinds of comorbid diseases can also make a severity towards the infections of this bacteria or these kinds of microorganism. So so most of the time, okay, this is a pillar of the clinical decision making. So as I mentioned just now, the first clinical decision is based on the diagnostic, based on the syndrome. Then after that, we will go for prognosis. The prognosis will identify the etiological agents in terms of the what kind of a species does it contain, does the bacteria or the parasite has a capability to have a resistant genes inside have a what resistance ability, if it resistance, what kind of drug resistant to okay, in terms of and after that, does it has in terms of the host itself is a comorbid? Is there is a patient has other illness like ages? Okay, if this too old, that means that the immune system is very weak. Okay, if there is a diabetic, if there are failures or the blood pressures. Okay, so this is kinds of the prognosis that we're going to do after the diagnosis. All this kind of information will lead into the treatments or give of the management of the patient after that. Okay. So just to highlight again, what is prognostic or prognosis? Prognostic is a predictions of outcome of disease based on the pathogenesis of the disease and the presence of risk factors for the disease. So what is the risk factors here? Presence of the risk factors here. Okay, so prediction, you know, that we presume that when this come, for example, someone like for example, methicillin resistant Staphylococcus aureus, okay, we know that there is a resistance towards a drugs, okay, towards multi drugs resistance, okay. So we know the outcome of the disease will be a severity, okay, the severe of the symptom, okay. Pathogenesis of the disease, we know that the Staphylococcus aureus can cause a respiratory tract infections, okay, and the risk factors is a failure, organ failures, perhaps the virulence factors such as production of toxin can cause apoptosis. The risk factors also can due to the high infection, high rate of infections, okay, like COVID-19, like HIV, high amplifications of the viral load, very high, so infection or the transmission is very high, so it can cause to the severity of the disease to the patient. So if I can say that, I can conclude that prognostic is actually is a so-called, it also can be called precision medicine. Okay, precision medicine, it means that it's very particular towards the host itself. Okay, we are identify the agent that cause to the host sickness. Then after that, we also monitor the host itself. Either host has a multiple illness, or we also identify either the host has a susceptibility towards to receive the infection, or does the host has no capability to receive the infection. So this is kind of precision medicine. It can be either population precision medicine, or it can be individual precision medicine, because a certain races, certain group in the world, like European, in Asian, they have their own genetic markup or capability to receive infections, like susceptible to tuberculosis, susceptible towards HIV virus, COVID-19. So these kinds of genetic markup, or the environment play a role in order or the risk factors that can play a role to predict the severity of the disease towards the population and also towards the individual. I also can presume that prognostic also for infectious diseases can be a molecular epidemiology studies, okay, because we are using a kinds of the use of omics, okay. We can use the use of omics, we can do PCR, we can do qPCR, next gene sequencing, in order to identify the susceptibility of the host itself, or we can also identify what is the variance of the agent, like dengue. We can do qPCR to identify what subtype of the dengue that infect patient, either is type 1, type 2, or type 3, or type 5. Okay, so when we know the subtypes, we know that where the origins of the virus itself. Okay, so when we know the origin, we know how to treat the patient itself. Okay, so for prognostic of infectious diseases, okay, this is kind of prediction risk of infection by investigating, as I mentioned, prognostic also can be a molecular epidemiology and also a precision medicine. Okay, so it predict risk of infection by investigating epidemiology of disease. Okay, it can be a population or it can be individual. The etiological agent, the agent itself, it can either bacteria, it can be virus, or it can be parasite, and host susceptibility. The host susceptibility, it depends on the host itself. Is it infant? Okay, when it's infant, they will be more susceptible to the infection, and also the old patient, the old people also very susceptible to the infection compared to the middle ages, because your immunity is very strong compared to those population infant and also elders. Okay, so when we talk about this prognostic or infectious diseases, you can see that there is a correlation between the epidemiology, etiological, and host susceptibility. Okay, so if you can remind or you can revise back your TFM 501, so can you tell me what is the correlation between these three towards infectious disease? Anybody? Anybody in the class? You can just shout, you can just open your mic. Okay, so how you correlate this prognostic with the TFM 501? Okay, we already learned this. Okay, anybody? Ian, the class rep?
Yeah.
Can you relate these three: epidemiology of disease, etiological agent, and host susceptibility? Can you relate this with the infectious diseases? What are the things that we learned already in TFM 501 that correlate these three for infectious diseases?
For prognostics,
Not for prognostic, for infectious diseases.
how you mean how do we correlate epidemiology
Yeah.
of the disease, etiology, and host susceptibility to
Yeah.
to what?
To infectious disease.
Oh, so epidemiology is basically how this infectious disease live around us. And then etiological agent is the infectious disease that cause the disease, and host susceptibility is the ability of this host to get this disease by this infectious agent, something like that.
Do you remember the terms that we use in TFM 501?
You mean what term?
What term that we use for this correlation? Anybody? Go? Right?
The triad?
The triad, yes. The
Yeah.
triad. Okay. You can relate the prognostic of infectious disease with the epidemiological triads. Okay. This is epidemiological triads, which correlate the agent, the etiological agents, the host itself, and the environment. The environment is the epidemiology. Okay. The environment, okay, and also when we talk about prognostic, the time frame of the patient goes to the hospitals also play a role for prognostic purposes. Okay, so please remember that this epidemiological triad is a very important either to look at the epidemiological of the disease and also for prognostic as well. Okay, and also diagnosis also can be ruled out from this epidemiological triads. Okay, so back to these things. Okay, so back to the prognostics. Actually, how prognostic play a crucial role in reducing the burden of infectious diseases. Okay, so we already know that prognostic is prediction. So how can prediction play a crucial role in reducing the burden of infectious diseases? Okay, so the first one is early detection and prevention. So when we do the prediction, when we know that there is a presence of the risk factors, so mean that we know that okay, we can do a prevention like dengue or like HIV. We know that all these fellas or the host itself has some susceptibility toward HIV virus. Okay, so for prevention, you have to consume some drugs that can inhibit the transmissions or insertions of the HIV virus into your body. Okay, some is like tailored treatment strategies. Okay, it means that if you have been infected with the HIV virus, how come we going to do? How we going to treat the host itself? So it means that we need to provide them with antiviral. Okay, we need to provide them with the inhibitors in order to avoid the amplification of the virus DNA RNA into the chromosome of the host. Okay, and also the prognostic can also play a role in order to devise a vaccine. Okay, vaccine development also can be devised based on the prognostic. Okay, because we know that the transmission is very fast, the rate of transmission is very fast. So we know that what type of the like COVID, for example, we know that type 1, type beta, type gamma, type alpha. Alpha is much more prone, highly infectious because there is a mutation of the surface protein, the S protein of the COVID. Okay, so because of that, we devise a vaccine based on the S protein, So it means that there is no insertion or there is no interactions of the virus with the receptors on the mucosa or in the nose or in the throats, okay, in your mouth. So we can devise a vaccine or we can devise a therapy based on the prediction itself. Okay. So as we mentioned that earlier, that we're going to use omics for this kind of topic. So when we call omics, there are multiple omics that we're going to use. Okay, so kind of genomic. Okay, the omics that we use DNA, omics that you use protein, we call it proteomics. Omics that we use RNA and mRNA, we call it transcriptomics. Okay, phenomics, okay, this is phenotype morphological of the agent itself. Metabolomics, okay, that we call to study a metabolites profile of that produced by the agents or metabolites that are produced by the host itself, intermediates, hormones or other signalings that related to the metabolite. So we can use this prediction, but most of the time, these kinds of omic are interrelated, or it can be integration. It can be used as a solo indicators. Okay, so most of the time, when we talk about integrations of omics in the infectious disease, there will be integrations of omics. Okay, integration of omic based on the host pathogen interactions. Okay, host pathogen interaction. So for prognostics for infectious disease, it can be produced based on the host pathogen interaction. It can be based on the host itself, and also it can be based on the etiological agent itself. Okay, so it based on this kind of genomic. It's based on DNA, RNA. Okay, most of the time genomic is DNA lah. Okay, transcriptomic is on RNA. Proteomic is on protein. So we will play around with these trees. Okay. Other than that, will be additional input from in this class. Okay. So I just mentioned just now, prognostic is mostly related. Prognostic or prognosis for infectious disease is related to the epidemiological triad, which is interrelated with the agent itself, okay, the virulency of the agents, the host, the genetic markup, the susceptibility of the host, and the environment, the epidemiology, okay. So for the infectious disease, if it's related to vector, then there will be a vector inside here, okay. But for this, for prognostics, because we are measuring the host impact, so most of the time in the environment is epidemiology, is the presence of the facilities, the time frames of the patient goes to the hospital. Also can predict. We can predict on the life expectancy of the patient. Okay. So this is a kind of diagram, okay, that help you to understand on the epidemiological triads. Okay, so environmental factors I mentioned just now, either access to the healthcare, timing of the sepsis therapy, a treatment initiated. So when you've been infected, how fast the sepsis can be. So this is an example of the sepsis. Okay, so bacterial infection. So how fast the sepsis been onset. So how fast you provide the antibiotic to the patient. Okay, it is because it will lead to the impact, the effects of the sepsis to the patient. Okay, clearance of the bacteria, the severity of the patient, the severity of the disease to the patient. So it will related to these kinds of environment factors. In terms of pathogen vector, there is a variant? Is there virulence factors that have been produced? Is there any resistancy presence on the individual bacteria? Is it co-infection and synergism between pathogens? Is there any secondary infections? Okay, so these kinds of pathogen factors can be correlated with the severity of the patient disease into the patient. For the host factors itself, genetic predisposition, comorbidities, okay, factors such demographic, social demographic, in terms of age, sex. As I mentioned, ethnic origin also play a role on the host factors. Host prognostic factors. Okay. This is the additional information that this is related to the metabolomic, because in our body there is a presence of microflora. The microflora is regulate our body in term. It helps to increase the immune system, increase what we call it guard. It will guide our system towards the other infection. Okay, so we also need to know the diversity of the microbe. Okay, so if you are susceptible to TB, tuberculosis, probably because the microbiome in your body is less or different between others. Okay, so that's why you are very susceptible to tuberculosis infection. Because this is due to the microbiome modulate the host immunity. Okay, so for this example, okay, perhaps for this patient A, and if day one they come with symptoms like fever, but when you provide antibiotics to this patient, they already clear at day two. Okay, there is no fever, there is no symptoms. It means that the clearance of the bacteria already been done. Okay, at patient A, due to the any factors, because perhaps the immune system very high. Okay, because the pathogen itself is, there is no virulence factors produced by the bacteria itself, so it's easy for clearance of the bacteria. But for another populations like patient B, it took around 5 days to clear all these sepsis, to clear all this bacterial infection. This is perhaps due to this patient perhaps has a commodities. They has diabetic, so perhaps the immune system very low. When there is a infection, even though it's not very virulent, but because this immune system very low, it will take some time when you provide the antibiotic. So it will take some time for the patient to recover. Okay, so this is kind information that related to if you want to look at associations of the omic itself, like serum biomarkers, transcriptomic, okay, non-coding RNA, proteomics, metabolomic, and epigenetic, that perhaps can be used as a molecular phenotype biomarkers that can use in order to look at the interrelation of these triads. Okay, so then we will go further one by one for host factors and pathogen factors. Okay. For host factors, understanding how human genetic or host genetic variation influence infectious disease susceptibility offer the opportunity for new insight into pathogenesis, potential drug target, risk stratification, response to therapy, and also vaccination. So I think you already understood with a very clear understanding on the based on these triads and also the factors that are related to the host factors, which come out with this definition. Okay, especially on the how the host genetic variation or how the can influence infectious disease susceptibility. Okay, and which when we know the genetic variation, that it will lead to the devising on the therapy and vaccination towards the host. So that's why I mentioned that prognostics of infectious diseases, it can be a precision medicine, because when we know the genetic markup of your individual, we know that what we're going to target, or we can predict how long the disease can be onset before it become more severe or can be clear. Okay. So With the help of technology that we already have now, the Genome-Wide Association Study, GWAS, or by using next-gen sequencing, has highlighted a few studies and a few examples that how host factors can correlate to the infectious disease and how actually the genetic markup of the host itself can be predicted for the severity of the disease. For example here, okay, so for HIV or AIDS disease, okay, so when we screen the patient, there is a certain markers, certain genetic markup, genomic, okay, so with this is genomic, DNA or gene that we screen, such as or proteomics, we search or we screen, such as CCR5 protein, okay. So what is CCR5 protein? Okay, so let me show you. So if you go to your eLearn, so there will be a folder of references. Okay, let me show you. So in your eLearn, there will be a references here. So if you click the references here, I already deposited a few references here. Okay, so 1, 2, 3, 4, 5, 6, 7, 8. Okay, so from these 8 references, I create the examples for your understanding. Okay, so for HIV/AIDS, if you can go to okay, if you go to topic or references called host genetic variation and HIV. So for page, if you go to page 4, okay, if you go to page 4, you can look at the CCR5, okay, so let me check. Okay, you can page 3, you can look at the CCR5. What is CCR5? CCR5 is a chemokine receptor physiological responses for binding of blah 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 blah, for which affect HIV acquisition rates and disease progression rates. Okay, so as we mentioned that host factors can be screened or can be examined for the prognostic of disease, because we want to look at the disease progression. Okay, so based on these slides, we saw that for HIV/AIDS, for host factors itself, CCR5 play a role in order to make sure that there is any severe progressions of infection, HIV infection, or there is no infection for HIV virus for this individual. Okay, so if you look at and you read more, this one say that homozygosity for a 32 base pair deletion in the coding region of CCR5 or CCR5 delta 32 accounted for reduced HIV infection risk. So mean that the population or individual who has a CCR5 delta 32 probably has is not susceptible or resistant to HIV infection. Okay, and look, if you read more, the frequency of the variant is the highest among people of European descent, in whom 10% of individuals carry a copy of the CCR5 32. So what we can predict, what we can presume, that European people has what? Frequency of the variant is the highest among people of European, mean that HIV virus infection is less compared to the Middle Eastern and African, because it said that intermediate among those of Middle Eastern and Asia are near absent amongst individuals of African ancestry. So it means that people with CCR5 delta 32 has possibility has opportunity to have no HIV infection compared to people in African or in the Asia, in the Asian, okay, which has a prone towards HIV infection. So when you do the prognostic, when you screening the patient itself, when you screen the patient, you screen for the CCR5 variant. Okay, you see the CCR5 variant. Okay, and when you do the next gene sequencing, you look at the it has a deletion of at the position 32 or not. If there is position at 32, you know that this individual has no resistance to HIV. Right. So we can predict that, even though the mother or the sibling has HIV, but probably the prediction to this individual is that this patient probably has a zero probability to get infection with HIV virus. Okay. In terms of severity, perhaps even though there is an infection, but the severity will be less because the infection will be terminated soon, because CCR5 is important to bring the HIV virus into the macrophages, or in order for the amplification process. Okay, so another one is a human leukocyte antigen HLA class 1 alleles. Okay, so for HIV/AIDS virus, most of the time they are using HLA MHC class 1. Okay, so if you look at the paper here, so it's also mentioned that cytotoxic T lymphocyte, also known as HLA, okay, is to recognize target cells through virus-derived antigen presented by MHC class 1 molecules. Okay, so most of the time, MHC class 1 molecules are related to the HLA A, B, and C, while D class 2 HLA. Okay, identification of HLA specific for HIV raised the possibility that the highly polymorphic HLA class 1 region may be involved in modifying HIV disease on the basis that such variation may alter CTL recognitions. Okay, so other than that, you can read more. Say that HLA-B 57, 27, 58, 51, 13, and 81 are amongst the most reproducible protective correlate to HIV. However, HLA with BW4 has been reported to be associated with enhanced HIV viremic control and reduced disease progressions. So, I hope that you can read these papers in order to understand on these slides. Okay. So other than markers that we can do or monitor for HIV for host itself, is a CCL3-1, a potent human immunodeficiency virus suppressive chemokine. So you also can read on the CC3L1 here. So you can search around. Okay. Where is the CC3L1? Yeah, this one. Including CCL3-1, it shows that which have more been implicated in HIV control. So people with high rate of CCRL1, it has low susceptible to HIV virus compared to people with less CCL3L1 on the macrophages surface. Okay. So this is a macrophages, okay, CD4 related immune systems. Okay, so this is for HIV. Okay, so we go next. Okay, so for other example, tuberculosis. Okay, there is another susceptible gene related to the for TB infection, such as chemokine CC motif, human leukocyte antigen HLA class 2 alleles, okay, and also natural resistance associated macrophage protein NR1 gene. Look for the paper on the host genetic tuberculosis. You can read from page 1 until page 9, but I would like to highlight here at page 2 say that today many studies, because today many studies have shown that the evidence of an association between chemokine, macrophage protein, and most of these genes so most of the time for infectious diseases, most of the time the genomic for host protein of the host is related to the chemokine production or immune system productions. Okay. For in human, environment gene is expressed in macrophages. Okay, so you can read then where is the mutation of the NR and gene NR1 protein. There is an inhibition on the intracellular killing mechanism of M2-glucosidase macrophages. So there is when there is a mutation of NR1 of the macrophages, so there will be the inhibitions of intracellular killing mechanism. So the macrophages cannot kill the tuberculosis when they infect the macrophages. Okay, because the mutation itself. Okay. So people with this NR1, so they have a susceptibility towards TB infection. Other than that, if you are producing APC that produce HLA, okay, HLA MHC class 1 or class 2, they show that HLA study carried out in the Asian region show that especially in India, it demonstrated the association of HLA DR2, HLA DQ1 antigen with susceptibility to positive tuberculosis. So it means that India has a more susceptibility of TB compared to other regions because of the presence of the HLA DR2 and HLA DQ1. Okay, so you can read further in order to understand of this topic or this papers. Okay, just I'd like to highlight that for tuberculosis for host itself, there is a three major genetic or genomic marker or genes or protein related to the host itself. Okay. For agents, so definitely we're going to do phytogenomics. It's a discipline that seeks to mark out virulence factors. So we want to look at the genotype of the agents or the phenotype of the agents. So the genotype and phenotype is related to the virulence factors, which can contribute to overall pathogenesis by comparing gene repertoire of pathogenic and non-pathogenic species. So for genotypes, okay, we are looking for what variants of like dengue 1, type 1, type 2, type 3, type 4, or COVID, beta, alpha, delta, okay, most of the things, okay. Drug resistance, what kind of drug resistance? Is it based on what? Is it based on the pump itself, or is it based on the production of the beta-lactamase that can cause to beta-lactam cannot produce the effects? So this is kind of genotype that we look at the agent itself, the virus or the bacteria itself. Okay. So as this is an example of the HIV. So it shows that type 1 HIV is the most common type of HIV with more than 90% of HIV AIDS cases in the world. Okay, compared to HIV 2, HIV 3, HIV 5. Okay, so HIV 1 is most dominant, can cause to infection, and it's also cause high transmission compared to HIV 2, HIV 3, and HIV 5. Okay, so this is more common type. So it means that the genetic markup when there is a HIV 1, so we know that we can predict that the transmission or the infection will be high compared to if there is a HIV2 or HIV3. For phenotype, definitely we are going for virulence factors like surface proteins. Okay, does it produce a toxin, pump proteins that can help on the intake of the nutrition or influx of the drugs. Okay, metabolic products, toxin enzyme, or infectivity rates. Okay. So this is an example of HIV protein, HIV virus that which has a capsids and also surface protein. So most of the time we will go for GP41 when there is a mutation of GP41. Most of the time it will enhance the attachment of the virus to the host cells. And when there is a mutation that in the virus replication rate or reverse transcriptase enzyme, it can cause a mutation, can cause a slow replication of the virus inside the host cells. Okay, so this is a phenotype that we look at, but it interrelated to the genomic itself. Okay, so on host pathogen interactions, most of the time, so this is very important because when there is a pathogen impact, so definitely the most important one that will be triggered are immune systems. Okay, immune response. Okay, this is most important when there is infection. It's compared to cancer, there is no immunity being onset, but for infectious disease, definitely the immune system will be come first. However, how clever is the bacteria or the microorganism to invade or modulate the immune response? Okay. So for host pathogen interactions, so for example, when you go for transcriptomic approach, okay, so what kind of transcriptomic that you're looking at? Okay, so most of the pathogens stimulate inflammatory hosts, respond to produce pro-inflammatory, as I mentioned. When there is infection, the immune system is the first that will be triggered. So most of the time, we will go for gene regulation related to the pro-inflammatory cytokines. Okay, so the cytokine variable effector define mediator via massive gene reprogramming in host immune cell. So this is a technology that we can use in order to look at the inflammatory host factors such as single cells RNA sequencing, or we can use gene microarray in order to look at there is a RNA that related to interleukin productions, transcriptions, cytokine transcription, TNF alpha transcriptions. So we can look at this transcriptomic approach. This is example for tuberculosis. We found that when there is an infection, tuberculosis, there is about 393 transcript signature correlated disease as well as treatment response and was related in two independent patient populations. So it means that there is a transcription process activated, and they found that when two independent patients been accessed or been examined, there is about 393 transcript signature related to the tuberculosis infection in the host. For proteomic approaches during infection, there will be a tissue leak protein and other molecule into circulatory systems. They can become a good biomarkers and a good prognostic to show that how severe is the disease. When this is a procalcitonin, it means that the tissue already been leaking, been wounded, or there will apoptosis happen. So the procalcitonin is a markers. They show that there is a apoptosis happen in the cell. So there will be need a treatment for that. Other than that, cytokines, C-reactive proteins, okay, this is related for infection. So when the C-reactive protein is high, we know that the viral load or the bacteria is very high. Okay, so most of the time, the technology that can be used as mass spectrometry, because we want to quantitate these individual markers, okay, or nuclear magnetic resonance, okay. So such as for tuberculosis, they found that proteomic analysis have found several clinical useful signatures And one proteomic study using mass spectrometry generated a fingerprint that distinguished active TB from latent TB in symptomatic patients presenting to clinic. For metabolomics, it's comprehensively to specific metabolic profile in bioelectrical fluids can be used as biomarker of the disease diagnosis. Okay, so for example, for tuberculosis, there are about 20 metabolic signature distinguish patients with TB from healthy individuals. Okay, and for microbiomes, the use of microflora. Okay, while most molecular assays target only a limited number of pathogens using specific primer or probes, microbiomic approach characterize all DNA or RNA present in a sample, enabling analysis of the entire microbiome, as well as human host genome or transcriptome in patient sample. So most of the time, when you look at this, the technology that we can use is a NGS. Okay, this is an example for tuberculosis. A gut microbiota play a significant role in modulating the host immune system. When there is a lack of the microbiome or species in your stomach, in your intestine, so the possibility for you to susceptible to TB is very high. Okay, as mentioned here, gut microbiota alternation were associated with the disruption of host responses against TB infection in the healthy populations. Okay, so as a conclusion, I say that pathogen detection assay has a highly variable sensitivity and specific rarely provide prognostic information due to high individual variability. As I mentioned, the integrations of the omics in order to look at the prognostic of infectious disease is quite challenging. Okay, quite challenging because the information in terms of the variability of the sensitivity and specificity of each of the technology can cause to the variable reading of the predictions. Okay. Measuring the host response fulfill a complementary role that address many limitations of pathogen detection assay. Omic technology have created opportunities to identify unsuspected and unbiased biomarker as well as biomarker panel and good for disease management, especially prognosis of the infectious diseases. So as I mentioned, that multi-omic integration for infectious disease investigation is include genomic, epigenomic, and also phenomics. This integration of multi-omics data is very complicated. Okay, you need a machine learning. You need a machine learning or AI, artificial intelligence, to help you to analyze all this data in order to correlate or to provide you a objective result for you to carry out the decision for the patient itself. So in terms of multi-omic challenges, especially on the heterogeneity, so because the technology that we use will be different, so the heterogeneity of the data will be available. So it is quite difficult to conclude either because of these factors we can go for this treatment. Because of this factor, we can go for this for device for the vaccine. So there need many study or database that needed in order to conclude of the interrelation of the omics in order to lead for the treatment. Computational complexity, you really need a very large scale data in order to train or manage the omics data. This is kind of big data. Okay, so you cannot have 100, but you need more than 1,000 or 2,000 data in order to train the machine in order to give you a decision to help your decision. Okay. Interaction and network, so there is lots of interaction when there is infection. There are lots of interaction, especially immune systems. Okay, constructing meaningful network or pathway from integrated data is very complex because not even in immune system, but also other signaling pathway that can be activated especially at the tissue level or cell level. Okay, so this is a multi-omic challenges related to the use of omic, integration of omic in the prognostic. But as I mentioned, omics it can be used for prognostics. Okay, it's not similar to the cancer cell itself, but because it's interrelated with the triad epidemiology, it's related to the host itself, the agent, and also environment. So I think without, okay. I think I end my presentation or my class today. So I hope that in the references in the e-learn, you will find this paper, Application of Omnic Technology to Combat the COVID-19 Pandemic. Okay, so this is 2021 paper. The one that I want to highlight here, I want you search for this kind of information, genomic analysis, identify mutation or variant of the SARS-CoV-2, okay, transcriptomic analysis, detect changes in SARS-CoV gene expression, detect changes in host ACE2 gene expression, and detect on dysregulation of immune response based on when the COVID-19 infection infect the host. Interoperatomic and metabolic analysis detect and quantify more protein and metabolites. Detect what kind of protein and how they quantify the metabolites or protein for the host itself. Okay, so I hope you can read this because this paper is very important because I will use this paper for your quiz. Okay, I hope that you can read this thoroughly. Okay. Actually, I already highlight some of the genomic analysis. This is variants that I found from the papers. Okay, and then after that, I already mentioned that the B1351 variant is a very highly infectious. Okay, this is due to the S protein mutation, highly transmissible, 50% compared to the variant, and resistant to convalescent serum and vaccine-induced serum. Okay, so hopefully you can identify more on this transcriptomic and proteomic. Okay, so please read this paper. Okay, so for that, I stop my class. Any questions?
Thank you, Doctor.
Okay, so all the information, the references will be you can find in the e-learn. Okay. So if you have problem, you just WhatsApp me. You can ask me directly. Okay. So if there is no questions, so there is a chat here. Morning, Doctor. Good morning. I think it's 21st. It's 28 got email. Today is DFM53. So this is definitely. I am not aware the date has changed again. Could you forward the message email to me? I have mistaken. Wait, let me check. Oh, okay. So I also not sure on the 21st or 28th. So I will check with the Dr. Fatma again on the quiz itself. I think 21st. I don't remember. But anyway, the quiz will be based on the e-learn. So we will deposit the questions there. So the quiz will be notified to you. They will open of the quiz for 1 hour. Then you can do your own. You can do the test. It's kind of not a test. It's a quiz. Okay, so you can open book. There will be open book kind of test. Okay, quiz. Okay. So without further, if there is no questions, so I can stop. So I can go for another meeting. Okay.
Thank you,
Thank you
doctor.
very much. Okay. Ian, anything you can WhatsApp me, okay?
Yeah, sure. Thank you, doctor.
Okay, thank you very much. Assalamualaikum. Bye.
Thank you.